Okay, folks, we will go ahead and get started. Good morning to everybody. I'm sorry to have to do this again. I took one of those at-home COVID tests on Saturday, and it was unambiguously positive. And then last night, I lost my sense of smell. Other than that, symptoms aren't bad. And by fall, with the CDC guidelines, I think I'll be okay to come back Thursday, perhaps Wednesday, so just stay tuned. In any case, thank you for your patience. At the beginning, I just wanted to point out that you have an exam starting in the testing center tomorrow, and it will go through the end of the day on Thursday. Please pay attention to the testing center hours and make sure that you know when the testing center closes. You may want to pay attention to line length. It would be a shame for anybody to be stuck in a long line and not have enough time on the test. The exam's 22 questions long. It's multiple choice. Most of the questions are worth 10 points. There's a few of them that are worth less than that and add up together. So overall, the exam's worth about 200 points out of 1,000 points that will be before the end of the semester. There's no time limit on the exam. You can write on the exam. If after the exam you want to see your work to figure out what you were thinking when you picked an answer, make sure to staple your scratch pages to the actual exam. They'll deliver those exams to me. You'll be able to see once the exam closes items missed and what the correct answers are. I will prepare a review sort of exam, post-mortem exam review for going through each of the questions and explaining that, and I will post that on YouTube once everybody's taken the exam. Let's see, other questions that typically come up. The testing center requires that a late fee kick in at 2 p.m. on the last day of the exam. So if you're going to take it in the evening on Thursday, it's going to cost $5. Please, please, please don't pay that. But they do that to stretch out the crowd. Because statistically, they know that about half of students will willingly pay $5 to have a little bit of extra prep time, and half will not, and that allows them to plan for the crowd. So I'm sorry for all of that. Anyway, anything else I missed? Anything else you want to know other than what's on the exam? The practice exam should prepare you well for that. Practice exam is not intended to mimic the exam in any way. Instead, it's meant to give you exam level practice on all of the difficult problems. Okay, so I will be looking at the chat. If you have any questions through today's lecture, go ahead and type them there, and I will try to answer them, and we'll just proceed from there. Okay, so let's see. Yes, I noticed the practice exam has a lot of nomenclature. Do we need to be super proficient in nomenclature? Not particularly. Do you get a note sheet to use on the exam? No. Can you write on the exam? Yes. Are there other options for taking the exam if you have COVID? Not immediately. If you're out with COVID, why don't you contact me via email and we'll make other arrangements. Today is the last day of material that will be relevant to the exam, yes. And I guess, Johnny, your follow-up question is, sometimes I mention a molecule and not the structure. In general, I'll show you structures. If I give you a name instead of the structure, it'll be on a fairly simple molecule. Abigail, what is the average amount of time students tend to take on my exams? Tends to be 2 hours, sometimes less, sometimes 1.5. Although you can, based on sort of your past history with test taking, you can scale from there. So I would say maybe 2 to 2.5 hours is good to plan for. More if your individual circumstance suggests that that would be good. Okay. So we're going to continue on with our discussion of amide bond formation. Last time I showed you 2 different ways you can take a carboxylic acid and convert it to an amide. Now we're going to learn how nature does that. And we're also going to learn a little bit about beta-lactam antibiotics. And then we'll see where we go from there. So this is the ribosome. The precise 3-dimensional structure of the ribosome is known. That's something that occurred during my scientific lifetime in the early 2000s. And what you see here are the 2 subunits, the 50S and the 30S subunits that come together kind of like a sandwich. And in between, you have places where the tRNAs can dock. So there are sites for the tRNAs. This is the exit site. This is the peptide chain site. In other words, the elongating peptide is linked to the tRNA here. This is A or tRNA that's T site RNA that's bound adjacent to the pRNA, and then this is a elongation factor that helps things move along, pushes the aRNA out of the way. Okay, so clearly there's a lot of this that I don't understand, and some of you actually may be better at this because of cell bio classes. But I wanted to talk just briefly about how the amide bond formation process happens in nature. So just for fun, I guess, we're going to draw the green tRNA. And thankfully, it's just going to be a bunch of squiggly lines. Oops, that's too big. All right, the tRNA has a shape like this. And down at the bottom here is the anticodon, 3 bases that are complementary to the corresponding codon on the mRNA transcript. And then the pRNA has a growing peptide chain attached to it via acyl phosphate linkage. So here is one of the phosphate groups on the end of the tRNA. The tRNA is made of nucleic acids. And then this oxygen is bonded to the first amino, to the N or the C terminal amino acid of this peptide. And then I'm just going to put R here for the rest of the protein. For the sake of our discussion, it's not particularly important for the exam or anything like this, but for the sake of our discussion, peptides and proteins are oligomers of amino acids. That is, I can draw actually what this R group is, what at least part of the R group is, sorry. Okay. Please understand that these R's here are for the amino acid side chains. This is one amino acid subunit. This is another amino acid subunit. They're linked together via an amide bond, which we sometimes call a peptide bond. Notice that there's directionality to this peptide chain. There's a nitrogen, an alpha carbon, a carbonyl carbon, nitrogen, alpha carbon, carbonyl carbon. So we call this end of a peptide or protein the N terminus, and this end we call the C terminus because of C for carboxylic acid. Okay, so this is what the RNA and the growing peptide chain look like in the P site. And important is this functional group here. This is called an acyl phosphate, but it's similar in reactivity to an anhydride. Which is where you have a carbonyl carbon attached to a carboxylate, which is a good leaving group. Only here, with the P site RNA, all of this is a good leaving group. Okay? Now, we'll go purple for the RNA that bears the amino acid. Okay, so this is the RNA that's bringing in the next amino acid. AA stands for amino acid. And this is also linked to the tRNA, to the purple tRNA via an acyl phosphate. And I guess I'll show things as they would be at physiological pH. That amino group would be positively charged. Okay, so you've got the growing peptide chain here. You've got the next amino acid here attached to the purple tRNA. What happens next is just straight out of chapter 16 chemistry. Some base would deprotonate the amino group to turn it into a decent nucleophile. Likely some hydrogen bonding groups are activating this carboxylic, activating the carbonyl to be attacked. The amino group attacks the carbonyl carbon to give you a tetrahedral intermediate. At that stage, your tetrahedral intermediate would look something like this. Oops. Then here's the nitrogen of your incoming amino group. And then over here you have your growing peptide chain. Squiggly line indicates that the protein is over there. Okay, this is a tetrahedral intermediate. You have the P site RNA and the T site RNA at this stage, and here is the growing peptide chain. I don't know if it's useful. We'll highlight the same things that we highlighted above. Probably we need a base to remove one of the protons on that nitrogen that attacked. Once we do that, lone pair electrons can kick down and kick off the tRNA as the leaving group, the green P site tRNA as the leaving group. And when that happens, we will have transferred our growing elongating protein chain from the P site, from the P green tRNA to the purple T site tRNA. And we will have made a new amide bond. Oops. So, you will have learned perhaps in cell bio that tRNAs are are in part responsible for reading the genetic code, right? The tRNAs bring the correct amino acid to the ribosome in a way that's complementary to whatever codon is present in the mRNA transcript. But what you may not have learned is that the other function of the tRNA is simply to be a good leaving group during the acyl transfer, during the nucleophilic acyl substitution reaction. Okay, so at this point, the growing peptide chain, and now a new amino acid that we brought in, and that new amino acid used to be attached to was brought in by the purple tRNA. So this process brings in a new amino acid, creates the new amide bond right there, and that process translocates or changes the position of the growing peptide chain, which used to be attached to the P site RNA, now is attached to the T site RNA. And then everything will just move down one. The P site RNA is released and exits here, and the T site RNA moves over to become the new P site RNA, and then space is available for the next tRNA to come in. Okay, that was a little bit convoluted, but I wanted you to see how protein synthesis, one of the most fundamental things in biology, is occurring through a nucleophilic acyl substitution reaction, which involves attack of a nucleophile on the carbonyl carbon, a tetrahedral intermediate, proton transfer to change who's the good leaving group, and then a leaving group leaves to give you a new bond. And this is the bond that we have made. This process occurs very rapidly. The growing peptide chain comes out of the ribosome sort of here. There's a tunnel that it passes through. And all of this happens extremely rapidly, many, many, many times faster than what we've been talking about here. So let me pause for questions. Let's see, what causes the whole thing to move over to the P site? I don't totally know. There are elongation factors that assist this process. This one, I think, pushes the A site RNA into the T site. I don't know what causes these things to happen. It's a good question. I'm sure it's known. I just don't know what it is. Other questions? Okay, let's see. Was this given as an example or something you should know? Well, you should have memorized all of the words I said over the past 10 to 15 minutes and be able to regurgitate them word for word. I'm sorry, I'm giving you a bad time. I apologize. I do want you to know nucleophilic acyl substitution. I don't need you to memorize the structure of a peptide chain. I would want you to recognize this connection, this acyl phosphate connection, as something that's similar in reactivity to an anhydride. So the basic principles of nucleophile attacks carbonyl to give a tetrahedral intermediate, which we can have proton transfer to change who's the good leaving group, then electrons kick down and leaving group leaves to give us a substitution product. That's all what I'd like you to be able to do. Recreate all of this from memory? Certainly not. Okay. Next, nucleophilic acyl substitution reactions give us a chance to talk a little bit more about what beta-lactam antibiotics do. So I'm going to scroll through a bunch of stuff here and actually copy the structure of amoxicillin. Some of you presumably at some point in your future are going to be prescribing amoxicillin to patients. And these are called, you may have heard of these as beta-lactam antibiotics. These are all penicillins. And the part of the molecule that's referred to as the beta-lactam is this four-membered ring. Just a nomenclature issue that you don't need to memorize, but a cyclic ester is called a lactone. A cyclic amide is called a lactam. So, and the reason this is called a beta-lactam is because you have a carbonyl carbon, an alpha carbon, the beta carbon, and it's a beta nitrogen that's tied back up with the carbonyl carbon in a beta-lactam. And this is a four-membered ring, and as you can imagine, it's highly strained. It's a four-membered ring fused to a five-membered ring, and there are a large group here, two methyl groups there. It's a sort of strained system. And beta-lactams are primed and ready to be attacked by nucleophiles. And the reason is, and generally amides are quite stable, right? I mean, we just talked about proteins, and proteins last a long time in biology, and that's because it's difficult to find a nucleophile that's strong enough to attack an amide bond. Amides are less reactive than esters because of, let's see, just a brief comparison. I think you've read this in your text, so hopefully it should be reviewed by now. But amides are less reactive than esters because the resonance structure where the nitrogen donates electron density into the carbonyl carbon to get that resonance structure is actually a better resonance structure than the corresponding one is for the ester. And that is because nitrogen is less electronegative than oxygen, and so is a better resonance donor. And remember, carbonyl reactivity has to do with the viability of this resonance structure where there's a positive charge on the carbonyl carbon. To the extent that this adjacent nitrogen can donate electron density into the carbonyl carbon, this resonance structure becomes more important than this one. And so you would expect amides to be less reactive than esters. Nevertheless, because of the strain, this beta-lactam is reactive and can be attacked by nucleophiles. So let's look at the biological mechanism for how this works, because it too involves amide bond formation. I'm going to show you some structures here. I will take notes on the structures themselves. You'll have access to these notes after class. It's not important that you memorize all the details, but I want you to understand the key chemistry that's going on. So beta-lactam antibiotics, the reason they are antibiotic is that they inhibit bacterial cell wall synthesis. Unlike our cells, bacteria have cell walls. So this is an example of, this is a close-up view of a couple of subunits of the bacterial cell wall. It's a complicated environment. You have long-chain carbohydrates, and you don't need to worry about these structures. Here's a six-membered ring. Here's the acetal carbon. Here's another six-membered ring. There's another acetal carbon. Two sugars linked together, and then each of them is part of a longer sugar chain. On one of the oxygens of one of these two sugars, you have a peptide chain growing off that oxygen. And I'll just try to illustrate the different amino acids that are part of this elongating, part of this peptide chain that branches off the carbohydrate. Oops. Let's just go, I already did pink, let's go green. Okay, so there is a peptide side chain that's linked to the sugar. There's the amino acid subunits labeled in various colors. And then one of the side chains of that peptide that's branching from the carbohydrate is itself linked to another peptide chain that has 5 glycine residues attached. Glycine is the amino acid with no side chain. And you'll notice that this 1, 2, 3, 4, 5 amino acid long chain is now linked to the end of a peptide chain on another carbohydrate. So let's just label this and get oriented as to where everything is. Okay. So you have one carbohydrate linked to a peptide chain. That peptide chain is linked to a second peptide chain, which is linked to the peptide chain of another carbohydrate. So this is a crosslink, and it provides structural stability. It's like a strut in a large skyscraper that links two different beams together and provides reinforcement, and this provides structural integrity to the bacterial cell wall. And this can go on indefinitely. See, this glycine chain from this peptide can now go and link to another glycan bearing another peptide chain. Okay? I'm worried that we're getting lost in the details, so let me pause for you to take it in and ask any questions you have. Okay, there's a interesting story. There's an interesting feature of this carbohydrate with the linked peptide chain. And a peptide and carbohydrate together are called a peptidoglycan. Okay, glycan for carbohydrate, peptido for peptide. The two amino acids on the end of the peptide chain from the peptidoglycan are both alanine, but they have opposite stereochemistry to what protein, they have opposite stereochemistry to the amino acid in proteins. Amino acids that make up our proteins all have L stereochemistry. Most of them have L as a archaic way of talking about stereochemistry. Most of the stereo centers in proteinogenic amino acids, that is amino acids within proteins, are all S. These stereo centers are R, but an archaic way of talking about it is to call them D amino acids. So these are two D alanine amino acids, and this is an important recognition point for the enzyme that catalyzes the crosslink. Look over here on the other side of the crosslink, we only have one D-alanine. So in making the crosslink, we're actually going to break, making the crosslink between this nitrogen and another peptidoglycan molecule, we're going to break the bond between these two D-alanine residues. Okay, so let's talk about how this happens. You've got, and we're going to focus in on making one of these crosslinks. We're going to try to figure out where this comes from, all right? So here is a peptidoglycan molecule that's not involved in any crosslinks. Here's your carbohydrate portion. Here's the branching peptide portion. And then here's the pentaglycine crosslink portion. All right, so the first thing that's going to happen is we're going to, sort of like we did with lipase chemistry, we're going to break the bond between these two D-alanine residues by using an OH group from an enzyme as a nucleophile. And so presumably some base removes a proton from that OH group, turning it into a good nucleophile. It attacks and we get a tetrahedral intermediate. I'm showing all of this at once. Presumably you could show the base removing the proton, then you get the alkoxide, then the alkoxide attacks. All right, so this is the enzyme that's catalyzing crosslink formation. Here is your tetrahedral intermediate. Again, here's the carbohydrate. Here's the peptidoglycan chain that comes off the carbohydrate. And here is the pentaglycine crosslink that branches off of that peptide chain. Here's our tetrahedral intermediate. And the enzyme is placed so that as these electrons kick down, the nitrogen can pick up a proton as it leaves. And what that's going to do is it's going to clip off, it's going to clip off this D-alanine and it's going to make a new bond to the enzyme. So let's see what that looks like once we've finished. Here is your carbohydrate. Here's your branching peptide chain. Here's the pentaglycine crosslinker. And now you only have one D-alanine and it's attached via an ester bond to the enzyme. Now in lipase chemistry, after we attached something to the enzyme via an ester bond, we had water come in and act as the nucleophile. In bacterial cell wall synthesis, the nucleophile is going to be this nitrogen, the terminal nitrogen on the pentaglycine crosslinker of another peptidoglycan. So this then acts as the nucleophile to attack the carbonyl carbon to give you a tetrahedral intermediate. And presumably there would need to be Proton transfer, let's see. Yeah, I'm going to skip this one. All right, so nucleophile attacks. Probably there needs to be proton transfer at the same time as there's nucleophilic attack, or you could imagine an enzyme removing one of the two protons on this amino group as the amino group attacks. We're going to sort of gloss over that. But ultimately, you get this tetrahedral intermediate where one branching peptide chain from the peptidoglycan is now linked in this tetrahedral intermediate via the Pentaglycin crosslinker in this tetrahedral intermediate, and at this stage all that's left is for the electrons to kick down and the enzyme group to leave as the alkoxide, and picking up a proton on the way out can then leave as the alcohol. And when that's done, we have our new amide bond highlighted in green. And again, that amide bond is between one of the nitrogens on the pentaglycine crosslinker and the branching and the peptide from one of the two peptidoglycans and the other peptide of another peptidoglycan. So this amide bond forming process makes the physical link between one peptidoglycan molecule and another. And notice that each peptidoglycan molecule has one pentaglycine crosslinker and one peptide chain. So this amino group could go on to react with another peptidoglycan. This peptide group could go on to react with another amino group, and you'd get an extensive system of cross-linking. Okay, I realize that's a lot, and again, I'm concerned that we're getting lost in the data, or lost in the details, so let me pause for any questions you might have. Instead of getting lost in the details, focus on the key points, which is nucleophilic acyl substitution. You attack a carbonyl, you get a tetrahedral intermediate, and the leaving group leaves. Questions so far? Tyler, you asked, in the tetrahedral intermediate, why was the breaking apart of those two D-alanines? That was, I think, at this stage. Why does the amide act as the leaving group? Why does this nitrogen act as the leaving group instead of the enzyme oxygen? You're absolutely right that you would predict the enzyme oxygen is a better leaving group than the nitrogen from this amino acid. I'd say the thing that helps this nitrogen leave is that it's going to be protonated on the way out. Perhaps it's even protonated first. So I didn't show you this, but it's possible that before it leaves, it has to turn into this, and then the leaving group can leave. That's probably more reasonable. Another thing that will drive this forward thermodynamically is just entropy, because you're getting rid of one amino acid. You're converting one molecule into two. Other questions? Right. So can I talk about how beta-lactam interferes with the pentaglycine cross-linking? Absolutely. So we're going to go back to the structure of the peptidoglycan by itself. Here's the carbohydrate, here's the peptide chain, and here's the pentaglycine crosslink. And it's going to come down to these two D-alanine residues. It turns out that as far as the enzyme is concerned, the beta-lactam looks like the amide bond in the beta-lactam looks like the amide bond between the D-alanine residues, amino acids. Now that's not immediately obvious, but if you had a three-dimensional structure of the enzyme bound to the peptidoglycan versus bound to the beta-lactam antibiotic, this antibiotic fits into that active site and puts this nitrogen, this amide bond, in the same spot as this amide bond would be. So at that point, the enzyme will attack, instead of having the enzyme attack the amide bond between the D-alanines, the enzyme will instead attack the beta-lactam carbonyl carbon. So let's draw what that looks like. Here's the enzyme group. Bonded now to the carbonyl carbon. That carbonyl carbon is bonded to oops, I think we got too many carbons there. So I'm abbreviating some of this structure for sure. Just want to make sure I've got everything in the right place. There's your tetrahedral intermediate. At this stage, electrons are going to kick down and kick off the lactam nitrogen. Again, you're going to think that's a terrible leaving group. Why should that happen? There's probably two things that make that happen. First, you have the strain relief that's going to happen when you pop open that ring. And second, there's probably some proton either from solvent like water or perhaps from the conjugate acid of our base in the enzyme active site that's going to protonate that nitrogen as it leaves. And when that happens, what have you got? You've got a now opened up beta-lactam antibiotic that is modified, that has modified the key nucleophile in the enzyme that catalyzes cell wall synthesis. The enzyme is now, we've acylated the key nucleophile, so it's no longer available to catalyze bacterial cell wall synthesis. In other words, if we go way back to the very beginning, if instead of this, we have the beta-lactam, if instead of having a proton on that oxygen, we have some other large group from the antibiotic. Now, there's no way that can act as a nucleophile to attack the carbonyl carbon, and so we can't catalyze cross-linking. That makes bacterial cell walls weaker and more leaky and makes it easier for your body to kill the bacteria. Okay, other questions? How have bacteria evolved to avoid this? Andrew, great question. There are a couple of different strategies bacteria use to deal with this. One of these is to prematurely hydrolyze the beta-lactam linkage so that the beta-lactam antibiotic never gets to the enzyme active site. Enzymes that do this are called beta-lactamases, and you can imagine them doing exactly this, only it's with a different enzyme, and it ultimately releases the hydrolyzed amino acid. So what I'm saying is some bacteria have enzymes that will catalyze the breaking of this amide bond in the beta-lactam into an amino group and a carboxylic acid. They'll catalyze the attack of water on this carbonyl carbon instead of the enzyme nucleophile for bacterial cell wall synthesis. So that's one. So there's resistance genes. Another general strategy that bacteria use is some of them have what are called efflux pumps, where they just take stuff that's inside, in particular antibiotics. They can take antibiotics that have diffused inside the bacteria and pump it out. And so those are a couple of different ways that they can prevent this kind of thing from happening. When you go to the doctor and say, I have an ear infection, when you take your kid to the doctor and say he's got an ear infection, one of the first antibiotics they'll turn to is amoxicillin, and they try to use that instead of a higher powered antibiotic because of the fear of generating even more resistant bacterial strains. And that's a whole area that I think we could talk about. The idea is that if you have, if you're treating bacteria with an antibiotic and you don't completely abolish the infection, all you've done is selected a population of bacteria that are somewhat resistant to that antibiotic. And so you can provide some selective pressure for resistance to the antibiotic to grow via the beta-lactamase mechanism or efflux pump mechanism or some other mechanism. And so it's a risk anytime you prescribe an antibiotic that if the patient does not complete the regimen until the bacteria are completely gone, you may be putting selective pressure on the bacteria to generate some kind of super resistant strain. So they'll start with these low power antibiotics because there are already bacteria out there that are resistant to them. So if it works, great, you didn't have a resistant infection. If it doesn't work, you haven't caused any trouble because there's already a resistant strain out there, and then they'll increase in power from that. Okay, other questions? George, you say, so because the enzyme reacts with the carbonyl on the beta-lactam, it has no way to detach as it would in the cross-linkage, so the enzymes get put out of commission. That's right. I suppose you could call the antibiotic some kind of perhaps a suicide inhibitor in the sense that the antibiotic itself is destroyed when it inhibits the enzyme, but the enzyme's destroyed too. I don't think, I'm not sure, I need to look it up, but I don't think there's any way for the enzyme to come along and hydrolyze this and rescue itself. And in that case, the only solution is going to be for the bacterium is going to be to degrade this enzyme and make more fresh enzyme that hasn't been modified yet. Okay, so there's some things that we haven't spent a lot of time on, but that I want you to know. I want you to go back and look at the reactivity of acid chlorides and anhydrides. It's very simple and difficult, and so we don't need to spend any time in class, but you should see in a fairly straightforward way how we're able to react acid chlorides with, say, amines or alcohols to get amides and esters, respectively. Same with anhydrides. Other than that, I think that's about it. So that will be enough for today, and I will hang around for another couple of minutes online if any of you have questions that you want to ask, and then I'll need to start the 10 o'clock lecture in a couple of minutes.